Hi, I have sent this question to you before. Now this is the solution for this question, right? Okay, we'll see the first part of the question. I think we have to see this picture. This is a white clove, and this plant is growing many parts of the world. Its scientific name is Trifolium repens. It's native to Eurasia, but is now a common plant found worldwide in lawns, next to roads, in pastures, and similar habitats. Some terpenes plants are able to produce the toxin hydrogen cyanide, HCN abbreviation, by cyanogenesis. Cyanogenesis is the production of cyanide. A study at 128 sites, 2,509 plants in Toronto, Canada, looked at the proportion of terpene plants producing HCN. The sites were at regular intervals from the city center towards rural area. I try my best to show this picture how it looks like when we are moving from a city center to the rural area. Here it's a crowded city with buildings, and then we get more nice and calm and quite beautiful village sites. That's how we can imagine how things are moving from city center towards the rural area. How this cyanide, hydrogen cyanide, can be toxic to the organism, so plants and animals and many living organisms, they depend on this electron transport chains and oxidative phosphorylation for ATP production. So if you recall the basic steps of this electron transport chains, which involve enzyme and also electron transport proteins. So this cyanide can block this cytochrome C level at the close to the fourth level cytochrome C. It can block the electron transport chain and reduce production of ATP, and this is a case for the death of the organism. Okay, this is the first set of data. On your y-axis, you can see proportion of plant with HCN. No units given, 0 to 1. And then x-axis distance from the city center from kilometers, so 50 kilometers is the maximum. And we can see the data scattered heavily. So this is the line, and state the distance from the city center at which the highest proportion of plant sample contain HCN. So these are the samples. That means one spot is a sample. So we can take this spot which has the highest concentration of HCN. So this how we can get this exact value on the uh, x-axis. So it is confirmed that this is the plant with highest concentration, because y-axis it shows like 0.9, more than 95, 97, 96, 98 around concentration, and this concentration corresponding to 32. So this answer is 32. Then the second question, outline the relationship shown in the graph. So in a graph, if we have this kind of correlation, there should be positive correlation, negative correlation, and no correlation. So here it is increasing. That means distance from the city center increase, the proportion of plant with HCN also increase. So then it's a positive correlation. So you can write answer as a positive correlation. Okay, this question is to determine whether similar pattern in cyanogenesis were seen in other cities. Studies were also carried out around New York City and Boston, USA, Montreal, Canada. So deduce whether the pattern of cyanogenesis was the same in all of the areas around all four cities. The previous one, we have seen it has positive correlation. New York, positive correlation. Boston, positive correlation. But Montreal, this has negative correlation because it's reducing. So that's a different, all three cities, they have a positive correlation, but in Montreal, it has negative correlation. Interesting. So this is the important thing and interesting thing as scientists. If you want to think, if all four cities they have a same pattern, a little bit easy to understand. There is a one factor which is same in all cities, but you should understand if there's a three cities positive correlation and negative correlation in one city, there should be a different, something different in Montreal, right? Okay, here New York City positive, Boston positive, Montreal negative. Now, how scientists think to address this kind of a question? Right? Okay, now you see three cities have positive correlation and only one city has negative correlation. So how you can solve the problem? Okay, now you get another set of data. Why you get this set of data? It's because of this. The researchers consider two possible ecological causes. So by looking at the observations, researchers made a hypothesis, and this is here there are two hypotheses, two assumptions. Why three cities it's positive, but only Montreal it's negative. First hypothesis is the researchers assessed the extent of herbivory of tea repens plant in Toronto by measuring the percentage of leaf area that was eaten. So let's say the plants are eaten by the animals. So if it's eaten by like this, the leaf, this much of, and when it comes to away from the city center, probably we can see more eaten by the herbivores. So we can see reduction. So the mean herbivory of tea repens. Here they take the percentage, and what we can see it is around close to 30. But when it comes to away from the city center, it's less than around 30%. The graph shows the result for both the cyanogenic and non-cyanogenic plants. Here they use the cyanogenic plants and non-cyanogenic plants. Discuss whether the data supports the hypothesis that the gradient in cyanogenesis is due to its benefits against herbivory in rural areas. It's like this. If a plant has toxic compounds, it protects eating by the predators or herbivores. So if the plants are toxic, they don't eat. So they check here whether presence of this cyanide is a reason that the herbivores not eat when it goes away from the city center. Okay, when we look these two data, the non-cyanogenic and cyanogenic, and if you look this the data which is scattered, They are largely overlapping. Almost everything is overlapping, and there's a little difference. That means if herbivores eat non-cyanogenic plant, same as the pattern for cyanogenic plants. So we don't see any difference because it's very close. There's a very little difference, and data is almost overlapping. So we don't see uh, that any big difference because herbivores eat non-cyanogenic. Similarly, they also eat cyanogenic according to this result. The answer is the graph data do not support the hypothesis because both show negative correlation between herbivory and distance from city center. There is little difference between cyanogenic and non-cyanogenic in herbivory. This difference is getting smaller between cyanogenic and non-cyanogenic in rural areas. Okay, the researchers then investigate the possible correlation between cyanogenesis and exposure to freezing condition. This is the second hypothesis. So they thought this difference is due to the temperature, freezing conditions. And it had been proposed that when a cyanogenic plant freeze, that's very important, its cell burst, releasing Hcn, which is toxic to the plant. So if it is the plant freeze, they are dying because of cyanide, because the cells burst. So evolutionary, they try to survive. So snow can insulate the ground and plants from freezing temperatures. If there is snow, the snow acts as a insulating layer. Like if there's no snow, the temperature is lower here. It's very lower because no insulation. So having snow is better. So here you see mean number of days below zero without snow cover. So here number of days is higher. So Boston has the city's area, the temperature is very cold because less snow cover and less number of days. Mean number of days below zero without snow cover. And here is the lowest. That means Montreal, they have a less days with the snow cover. That means they have a little bit higher temperature than these three cities. Toronto, New York, Boston, colder than Montreal. However, snow is more likely to melt in cities, which then exposes plant to freezing temperature. All four of the cities studied receive below freezing temperature and winter snowfall. Researchers look at the number of days below freezing and that did not have a snow cover in these cities. Identify with reason the city where the plants were more insulated from freezing temperatures. So we identified Montreal as the city because it has a less number of days with the below zero without snow cover, so less number of days. So we can take Montreal, right? Okay, using all of the data so far suggests whether exposure to freezing temperature in the four cities is supported as reason for the difference in HCl production in chili pens. Okay, if you look these three cities, Toronto, New York, Boston, they are cold because mean number of days below zero without snow cover is high. So only we can take Montreal, which has quite less temperature is a little bit higher than Toronto, New York, and Boston. Then it shows this negative correlation. So these cities, if you look, it is the 0. Here it's a 0. Close to 5, let's say. In the city, it's 0.5, and here it is less than 0.2, less than 0.2, less than 0.2. So Hcn concentration is less, and here Hcn concentration is high because the temperature is not that bad. They don't freeze, and cyanide is not released. It's not toxic to the plant, so they could have a higher HCl level is high. Right? The answer is cities with more days without snow cover have positive correlation between distance from city center and HCl. Fewer plants with HCN within cities that have more days without snow cover have more exposure to freezing temperatures. HCN is 0.2 or less. HCN in cities that have more days without snow cover proportion, whereas a city with fewer days, Montreal, it is almost close to 5. Yes, this is a very good question. I think this is a solution for your answer. I hope you can check your written answers with this one. And we will post another question next time. Thank you.